Hello, and welcome to week 2 of the ACT Prep Science questions. Let's look at question number 1. So if you look at question number 1 and you read it through, we're talking about photosynthesis in plants. It says that there's 2 different metallic paths for photosynthesis, designated as C3 and C4. Remember, photosynthesis looks at the fact that it takes in carbon dioxide to make sugars for itself, and it produces the waste of oxygen, so it gives oxygen off. It gives us these nice little graphs. This one deals with light intensity. This one deals with temperature. They both show the rate of photosynthesis when it does that. And we see the different types of plant. The straight line is C3. The dotted line is C4. The question wants to know, to test whether a given plant is a type C3 or a type C4, what measurement might be used. We wouldn't want to use, we have light intensity and temperature. And yes, those will produce different things, but look, light intensity at the same time is going to change around how much photosynthesis is going on. The temperature will change some of it. It'll change the type C4's photosynthesis that's going on. And so if you're using both of those to try to figure it out, you might be canceling each other out. You wouldn't want to use both at the same time. Glucose content of the leaves. Well, yes, you would be able to measure that, but if you're not doing it at different lights or different temperatures, then it's not going to make a whole lot of difference. It's not going to relate to C3 or C4 types of plants. Now, oxygen production over a wide range of temperatures. Well, now, if we're looking at just the temperatures, and we look at the fact that when we change those temperatures, photosynthesis changes dramatically for the type C4, but it doesn't change at all for C3. And note that when we're doing photosynthesis, we're going to get a production, or it's going to give off oxygen. So if you're measuring the amount of oxygen, you can measure the amount of photosynthesis at those different temperatures. And if it produces more at a low temperature and less at a high temperature, it's a C4. If it produces the same amount throughout all temperatures, it's a C3. So that would be a very good check. But let's look at the last one just to make sure. Rate of carbon dioxide production. None of them produce carbon dioxide. They take that in. So it is definitely C. And that's our answer. Question number 2. It's just looking at different pollutants in the U.S., and it looks at it in millions of metric ton, air pollutants. And so you have a nice chart, gives you the sources, and it gives you broken down in parts, millions of metric tons for the different oxides, oxides of sulfur, oxides of nitrogen, organic vapors, and carbon monoxide. The question, however, is looking at acid rain, and it's produced when you have water or rain mixing with sulfur and nitrogen oxides. So that pretty much does away with these two over here. We're just looking at oxides of sulfur and oxides of nitrogen. So it wants to know which of the following would be the most effective in reducing the amount of acid rain. Well, we can look over here and we can figure out what produces the most acid rain by adding these two together. If you notice, all 3 of these bottom ones are fairly small. And so like this would just be 0.1 total, 0.1 total. This one would be just 2.5. And transportation, that would be a 10. And industries would be a little over 12, and actually 13. 13.1. But then electric utilities, look, none of them even have come close added together to this 17.6. If we add that together with the oxides of nitrogen, then this is over 20. And so it's our big leader. So we're probably going to be looking for something that uses electric utilities. So insulation of buildings to reduce the amount of fuel heat. Well, that doesn't do with electricity, so no. And if we look at buildings, they're very low. Cleaning the stacked gases used in manufacturing. Manufacturing industries, that was higher, but nowhere close to electric. Conversion of car engines to use ethanol. Well, transportation is also the larger ones, but it's still smaller. And then the last, cleaning of stack gases used in electric power plants. Well, we need electric power plants for the electric utilities, and that's our biggest producer of the oxides that mix with water to make acid rain. So it obviously has to be J. Number 3. Number 3 is really a continuation of experiments that we looked at last week, just with a different question. So if you're confused about this experiment or how halogens work, you might want to look at that video. Let's see if we can figure out with what we knew from last week how we're doing on number 3. In experiment number 2, all the reaction vessels were dark brown. Yep, they all ended up dark brown. Well, it wants to know why. So, because. Well, we know from last week that iodine was the least reactive. And reactivity only deals with the halogen element itself. You don't use reactivity when referring to the salts. And so the first thing you can do is eliminate A and B because they're talking about potassium iodide salts. Potassium iodide salts. So we know A and B is not it because they don't have to do with the reactivity. Now, C and D are both looking at iodine and its reactivity with the 4 halogens. Well, if it was the highest reactivity, it would kick out the other halogens out of the salts. So we would get chlorine and bromine out of these, and you would get a different color. The reason it stays dark brown is because it has the lowest reactivity of all the 4 halogens, meaning it doesn't kick anything out of the salt. It just sits there and turns the solution a different color. And so our answer is C. All right, question number 4. It looks at different honeybees that are going on, and there's a disaster going on with the honeybees right now called colony collapse disorder. We're just going to call it CCD from now on. And so these 2 scientists are arguing about the difference with it. The gist of it is scientist 1 thinks that we don't know what the cause is. He lists a bunch of different contributing factors, but he says we don't know the cause. Scientist 2 says there's a bunch of different things listed as well, but there's a main factor, and that's insecticides. Insecticides are what he's all about. So this is looking at what both scientists agree. Well, viruses are the chief cause of CCDs. Would not be it because neither of them think that's the chief cause. The main cause of CCD is unknown. Well, number 1 thinks that, but number 2 thinks that it's insecticides. So they don't agree there. Many factors contribute to CCDs. Well, he said many factors contribute, and that's why we don't know what it is. He said many factors contribute to it, but they all come from this pesticide related stuff. And so, yeah, this looks like they do agree that there's many factors. Number 2 just thinks it's more towards one specific brand, the pesticide or insecticides. Insecticides are a chief contributing cause of CCDs. Well, 2 thinks that, 1 doesn't. So that was a no on D, no on B, no on A. C is in fact our answer. And so we're good to go. All right, last one. It's also looking at another experiment that happened last week. If you need more detail on that, go back to last week and it'll tell you some more about that. But this time we're adding something. We're looking at if they had another experiment with these 2 types of bacteria, R and Y. It says this time, because we've seen them in glucose, we've seen them in amino acid, we've seen them in glucose and amino acid, we've seen them by themselves and together in those different mediums. They say now they put them in one more medium, and that medium was ethyl alcohol and amino acids and glucose. But it says in that one, the R bacteria grew, but the Y bacteria did not grow. Now, it's a little tricky with what it's asking because it wants to know which of the following hypotheses could not explain it. So we're looking for one that doesn't make sense. Okay? So if it makes sense, it's not our answer. We're looking for one that doesn't make sense. So let's read it. ethyl alcohol is poisonous to Y, but not to R. Well, that would make a lot of sense. If it was poisonous, then it would kill off R, or it would kill off Y, but R would still be able to grow because it has glucose and amino acids that it needs to grow, and it just wouldn't deal with ethyl alcohol. So that makes plenty of sense. B, in the presence of ethyl alcohol, R bacteria produces a substance that is poisonous to Y. That also makes sense. Remember last week we talked about when R and Y are both put in glucose and amino acid, that Y produces something that kills off or keeps the R bacteria from growing. So it makes a lot of sense that when they're put with ethyl alcohol, it might do something in the same fashion. C, ethyl alcohol is necessary for the growth of R bacteria, but not for the Y type. That one doesn't make sense. So if you look at it, look, we've seen Y grow. We've seen them both grow in different substances without any ethyl alcohol. We've seen growth in all of these in 3 different experiments. So that completely discredits that one. Makes it absolutely nonsense. But let's look at the last one just in case. Ethyl alcohol prevents Y bacteria from making use of amino acids. Well, that would make sense because amino acids tend to be a major thing that's needed for Y to grow. And so that one also makes sense. However, C does not. C is our answer, and that will conclude this lesson. Good luck.